Mutations are mistakes in the DNA that can lead to an abnormal protein production. One type of mutation that we'll be talking about today is point mutation. Point mutations happen when one DNA base is substituted for another, resulting in a change in one codon in the RNA sequence. There are different types of base substitution, transition, and transversion. To understand transition and transversion, you have to understand that adenine and guanine are considered purines, cytosine, thymine, and uracil are considered pyrimidines. Transition means that you're going from either a purine to a purine or a pyrimidine to a pyrimidine, for example A to a G or C to T, while transversion means you're going from a purine to a pyrimidine or from a pyrimidine to a purine, for example A to C or T to G. There are 4 different types of point mutations: nonsense mutations, missense mutations, silent mutations, and read-through mutations. The following DNA coding strand and the corresponding mRNA strand will be used as the normal to demonstrate each type of mutation. A nonsense mutation is a genetic mutation that causes a change in a codon so it becomes a stop codon instead of an amino acid. This results in shortening of the protein. Comparing a normal and a mutant DNA, we can see that everything is the same except for the codon T, G, and C, which codes for the amino acid cytosine. After C was replaced by A, it resulted in a stop codon, causing the protein to be shorter. This is also an example of a transversion, since it's going from a C to an A pyrimidine to a purine. A mismutation is a genetic mutation that results in a change in an amino acid. This may cause an alteration in the protein function or can make the protein non-functional. Here, a cytosine replaced the adenine, causing the amino acid to change from the amino acid asparagine to the amino acid histidine. This is also an example of a transversion, since it's going from an A to a C, a purine to a pyrimidine. Since many different RNA codons can code for the same amino acid, a silent mutation can occur. A silent mutation is when a single nucleotide changes but still codes for the same amino acid, which may not affect the function of the protein. As shown on the screen, cytosine replaced thymine, but both codons still code for the same amino acid, the amino acid alanine. This type of change between the T and the C is also known as transition, since it's going from a pyrimidine to a pyrimidine. A read-through mutation is when a change in a DNA base causes a stop codon to change into an amino acid and continues with translation. This results in a longer protein. In this example, when we replaced adenine with guanine, we caused the stop codon to change into an amino acid, in this case the amino acid tryptophan. The replacement from A to G is also called transition, since you're going from a purine to a purine.